a este espacio del jueves de la biodiversidad, que es un espacio abierto a la comunidad académica externa al Instituto de Ciencias y que busca brindar información sobre temas inherentes a la biodiversidad. El día de hoy tenemos invitados a un grupo de trabajo de la Universidad de York, la Universidad College de London y la Universidad, los dos de la Universidad de Londres, ¿no? Sí. El doctor Ian Barnes, que es el líder de investigación en ADN antiguo del Museo de Historia Natural de Londres, la doctora Selina Brace, investigadora en ADN antiguo, y Juan Camilo Chacón, doctor y pasante postdoctoral de la misma institución, investigador también en ADN antiguo, y que nos visitaron en la colección de mamíferos y en Colombia en un proyecto relacionado con ADN antiguo en osos de anteojos, proyecto que se hace en colaboración con el Instituto Humboldt. Bienvenidos, welcome Colombia. I hope you enjoy your stay here, and always welcome.
Buenas tardes. Yo voy a como hacer nada más una introducción muy corta como el trabajo que hacen Ian y Selina, y luego también pues digamos voy a ayudar un poco al final de la charla si alguien quiere hacer preguntas en español y también como para traducir un poco las respuestas y todo eso. Entonces básicamente pues les cuento de qué van a hablar ellos hoy muy brevemente. Es como el cambio que ha habido en las poblaciones en la Gran Bretaña y de las discusiones que se han dado por mucho tiempo en la arqueología británica y en la arqueología europea acerca de cómo los cambios culturales de alguna manera se relacionan con simplemente difusión cultural o con reemplazos de la población. También Selina hablaría un poco más de la parte técnica de cómo el ADN antiguo ha revolucionado todo este tipo de investigaciones, y ya Ian nos va a dar más como una especie de recorrido por los estudios que ellos han realizado acerca de este tema. Entonces digamos que ambos tienen una experiencia muy amplia en el trabajo del ADN antiguo, y ya van a explicar muy bien de qué se trata. Entonces al final de la charla, ya saben, todas las preguntas que tengan yo las puedo traducir a ellos y asimismo las respuestas, y ya con eso digamos que de alguna manera quedan algunas dudas resueltas. Entonces le doy el paso a Selina.
Okay, good afternoon, and thank you for inviting us here to give this talk. It's a pleasure to be here. I'm not quite sure what Camilla was saying, but I'm still impressed.
Very general.
And I've come from London, UK, and I'll have to click that one. We all work at the Natural History Museum in London. It's a beautiful old building. It was opened in 1881, but the collection actually originates from the late 18th century. There are somewhere in the region of 70 million objects in the museum. A lot of them are actually beetles, but we actually have the world's largest collection of type specimens. So it's an excellent place to work if you're an ancient DNA researcher, which is what I am. What do we mean when we talk about ancient DNA, though? Sometimes this can be a bit confusing, because we're not really talking about time here. What we're talking about is any kind of DNA. Well, ancient DNA can be thought of as any DNA that wasn't stored specifically for the purposes of DNA analyses. So age isn't actually the important issue here. We work on material that is anything from tens of years old, hundreds of years old, to tens of thousands of years old. It's actually not the time, it's the environment that the sample has been kept in that is really crucial to our investigations and why we need to use special techniques. And the main thing we have to remember is that heat is not the friend of DNA. Heat breaks down the DNA at a faster rate and makes it into these much smaller degraded pieces, and whereas a cooler environment preserves the DNA. The kind of material that we work with is things like mainly bone, but also tissue and hair and teeth. but we often work with insects, so insect legs, sometimes bird toe pads, and then coprolites, and anything that was preserved in spirits. So why do we conduct DNA analyses on museum material? Well, one thing that's useful is it allows us to access certain locations over a time series. So museums often have collections where someone has gone year on year to the same place. So it gives us this elongation of time from one location. It also allows us access to material that may be very hard to find. It might be that it's from a very remote area, that it will be very difficult to go to collect the material, or it might be that the animal, such as in this case here, was actually extinct. So the only place you can go to is the museums to find this kind of material. So we work on a lot of different projects, but the kinds of questions that we can answer, we answer questions to do with taxonomy and evolutionary relationships. So this is quite often useful with extinct species. We want to know what they're related to today. We want to know what they're most similar to, or what family they belong to even. We want to know, or we can ask questions about past population history and population structure. So we might want to know something about a migration event, or a source population, or maybe see if there was a bottleneck in the past. We can also be very targeted, and we can go to very specific points in the past, such as if we know there's something such as a domestication event, we can actually go and sample material before the domestication event, during, and then after. And this then allows us to really zoom in on that time period and answer questions about how the genome was affected by whatever event we're trying to test. And we can also look at things like changes in allele frequencies, or we can ask questions about phenotypic and functionally relevant traits. Now this last one was actually only able to do fairly recently, because there's been massive changes in the way we do our work. It's changed radically over the last 10 years. And this is an excellent example. So this was in 2015, and we had, not we, the ancient DNA community had to, actually I think the entirety of the ancient DNA community are actually authors on these two papers, except us.
Yeah,
we're on there, so these two papers came out back to back in Nature in 2015, and they were both looking at this Bronze Age, and they were looking at human migrations. So it was a really interesting time, and suddenly we're looking at these two papers again, and they're looking at huge amounts of samples. They're looking at 100 individuals, 100 whole genomes from across Europe, from this sort of like 5,000 to 1,000 years ago. And the second paper was looking at nearly 70 individuals. This was SNP data rather than whole genomes. But it was hundreds of thousands of SNPs. So this is big data sets. And if we think to put it in an ancient DNA context, 10 years ago, we were working on, just a few hundred base pairs of mitochondrial data, and we were publishing on 40 samples at most, and maybe 5 authors. So it's big changes that have occurred fairly recently. What we can do with this data now, again, this is quite radically changed. We're now looking at things, principal components analyses, PCAs, it's very common in biology, obviously, but we couldn't do that with the data before. Now we can. We can be looking at every individual's ACGs and Ts and crunching these down into these two the principal components and looking at these to actually say something about these individuals. This is a visual representation, how closely related, how similar they are to each other. And what you can see is that peoples from, say, time points are grouping together. People from similar locations are grouping together. And it starts to form this pattern. The coloured ones are all ancient individuals, and the greyed out ones are actually modern individuals. It also allows us to look at things such as admixture proportions. So when we're looking at admixture proportions, we're taking an individual's genome, And we're looking at that in terms of where the source populations, as it were, for those genomes have come from. If we think about Europeans are descended from 3 major migration events. So their genetic makeup is made up of people who were hunter-gatherers, people who were farmers, so these are people from the Neolithic, and people called the Yamnaya. Now the Yamnaya people come from over here. So this is a region between the Caspian and the Black Sea, and they were like these nomadic herder people, and they were a highly successful group who then moved across Europe during the Bronze Age. But we can tell about this, and we can examine this further by looking at the genetics. Because so this here is one of these admixture plots, and at the bottom we have the ancient peoples, and this is through time. So this is the oldest individual here, moving up to more recent individuals at the top. And these colours, so the orange is represents the farmer ancestry, the blue represents hunter-gatherer, and the green is Yamnaya population that I'm talking about. So when you look at these oldest ones in the Neolithic, these are just farmers. The proportion of their genome can be seen to be shown is farmers. And then as you move up through time, you hit the Bronze Age, like here, the late Neolithic Bronze Age, and you see that individual's genome is completely taken over by Yamnaya ancestry. It becomes really green all the way across, and then this is just reflected all the way through time. And this is interesting when you look at modern data. So these are modern day individuals who've been sequenced, and when you look at them, the proportion of their genome that could be represented by these 3 types forms this north-south cline. So the most northerly individuals, this is Norway, Lithuania, Estonia, have the largest amounts of Yamnaya ancestry there, and at the southern points you have very, very little. And what this shows us is that the migration event would have come across in a northerly direction. So those southern areas in Europe were hardly touched by the Yamnaya ancestry. So I hope that makes sense. The reason for these changes has been next generation sequencing. This is what made the biggest difference. This is why we can generate all of this data. We can do this now because it's got a lot cheaper. To begin with, it was ridiculously expensive, but as you can see, its prices have dropped, and so, since 2007, 2008, and this is the latest was in 2011 on this graph, but yeah, it's now super cheap to do this. But using next generation sequencing still has its problems. We still have issues with this. So we've gone from a PCR-based analysis to next generation sequencing analysis, but it still comes with problems. So the first of our problems that we have is this one, this short fragment size. So this is what I'm saying, ancient DNA, as soon as anything dies, the DNA begins to break down, and it becomes these small fragments. Typically around 65 base pairs. But this then creates a massive problem when you're trying to de novo assemble. You're trying to put these pieces back together again. It makes that very difficult. It's a hard puzzle. We also have a problem with low complexity. So low complexity, what we're trying to do there is find the entirety of the genome. And you might find because it's fragmented, you've got a bit from that section, a bit from there, a bit from there. But you don't want that. You want the whole picture. So and it needs to be replicated, you need high coverage. So this can be a problem. And then finally, we have a problem with low endogenous content. What I mean by this is when we're sequencing, when we're shotgun sequencing, we're sequencing everything that's there. And when you're dealing with these ancient DNA, anything from tens of thousands of years old material, what you find is that you mainly sequence soil bacteria. You're not sequencing the organism you're interested in, you're sequencing everything else. And that can range from 0 to 99%. So how do we deal with these problems? In terms of the fragment length, it's a lot easier if you start working with something with a reference genome. It just makes your puzzle a lot easier to put back together again if you can use this, your reference genome, as something to align all your small fragments of DNA to. it makes it easier to put the puzzle back together. In terms of low complexity, what we're starting to find is if you do multiple extracts from the same sample, also if you build multiple libraries from the same extract, you can still pick up more and more sections of the genome. So repeating that over and over actually really does help. And in terms of low endogenous content, we can do things such as capture array, and with this we're obviously then just targeting the DNA that we want and pulling that out first and then sequencing that. But to do capture, you obviously, it's a bit circular because you do still need to know the reference genome in order to design baits and to work out what you want to pull out of the DNA to sequence. Another way that we found to enhance the endogenous content is sampling from the right material. It turns out that the Petrous Bone, which is the inner ear bone, so if you take a skull, actually, yeah, take this skull for instance, and you kind of like turn them upside down, you can see inside his inner ear bones here. These are really, really dense bones, and this really protects the DNA inside of that material. So it means it doesn't break down as fast, so you have longer pieces of DNA, and it also prevents any other sort of bacteria and other things getting into your sample. So you have higher endogenous content if you use the Petrous. Just point out, this is the this has actually been sampled. This is the inner aureal meatus. It's actually this tiny little dot here. It's our sampling drill hole. Just to point out that we sample only a very small amount from any of these individuals. We work for a museum, so it's very important to us that we don't damage our samples, as I'm sure Hugo can attest to as well. So this is a really good bone. It's not a silver bullet. You know, it's not. You can't always sample a Petrous and go, oh, it's always going to win. It will be marvellous. No, but if you try these, it does seem to work very well. And this is just a little example from just some of the stuff that we've done. If you get the very high, or very high, over 40% endogenous content, it's more likely to come from a Petrous than it is from anything else. So, yep, that's just that's the background to ancient DNA done, and I think Ian's going to. I know Ian's going to talk to you a bit more about what this project.
Actually, yeah.
No, go on. I'm done.
Okay. Yeah, so just to follow on from what Selina's been talking about, that's sort of an overview of the kind of work that we do. And so I thought I'd follow on by talking about two particular studies that we've conducted that were published in the last year, although they actually took us about the last 5 years to do, yeah. So both of these relate to people moving into Britain, into the British Isles, over archaeological timescales, so over the last 10,000 years or so. And I can imagine that's probably not entirely that relevant for this audience, but I had the slides, and that's how it is. But it's worth noting that although there's a long time scale and a particular set of questions here, the techniques that I'm going to talk about are the kind of things that we could use in a similar context here in Colombia, or over short time scales like the last century or so. Okay. So the other thing I wanted to remember was that Britain's quite a long way away from here, although I did notice that Bogota is looking very much like London today because it's raining, yeah. So it's just worth sort of putting this map up to make the point that why is Britain interesting? It's a rather small island on the edge of a much larger continent, and so there's not really it's not immediately that obvious why it's interesting. It's interesting for us because we're British, but also because it's the kind of end of the line for any migration processes that happen. So we've got a couple of examples here of people migrating across Europe and ending up in Britain. And it's that has been kind of an interesting element to the work that we've done for these two papers. So the first one of these was a paper that covered not just Britain, but actually large parts of Europe as well, and looks at a particular material culture, the beaker culture, which we'll have a closer look at in a moment. So it's a very large author list, and if you look very closely, you can see that Selina's there. Number 2 on the author list. I'm down, where am I? I'm there, excellent. So 3 from the back. So we did quite well there, wasn't too bad. I think it's 100 and...
50 odd.
Yeah, actually, yeah, I'm not sure you can count. I can count that way, so yeah, I go. So yeah, okay. Okay, so around about 4,500 years ago, 4.7 thousand years ago, we begin to see this material culture initially in Iberia, in the Iberian Peninsula, and then it spreads across Europe, first into kind of France and Central Europe, and then is carried north and northwest from there. And it takes, about exactly 400 years, 400 to 500 years to make that spread. So we start to see people being buried with this very characteristic pottery vessels, which are referred to as beakers. So rather than the kind of beakers that I'm used to, which are like the size of a glass, these are about this big, quite large in size. And it's not only do we find these beakers, we're at the same time finding stone arrowheads, copper blades, axes and daggers, these characteristic stone wrist guards for archery, and I'm trying to think if there's anything else, these V-perforated buttons that I'm sure is an example. So the paper that we're a part of reported 400 new ancient DNA samples, including over 150 from the UK. And so, just to put it in context as well, exactly what's going on at this time period, particularly in Britain. So this is a group of people who migrate into Britain kind of just after Stonehenge is built. So it's towards the end of the Neolithic period. Neolithic farmers have been in Britain for about 2,000 years at that point. And this new group turn up, initially not with copper artifacts, and then copper arrives shortly after that, within a few centuries. And this beaker phenomena exists for a few hundred years, and then kind of just dies out, just disappears. The data that we have in this paper, I'm not going to talk about the sort of the data generating process very much, but it's a combination of SNP capture data, that Selina mentioned before, generated in David Reich's lab in Harvard, and then also whole genome shotgun sequence data that we generated in our lab for the UK samples. So it's a kind of a merge of those two, with sometimes a lot more of the SNP capture data, and sometimes not so much. And we'll get there eventually, yeah. So, one of the really obvious questions, a really long-standing question in European archaeology, in fact world archaeology all the way through the 20th century, is about how material culture, like those beaker pottery vessels, actually spreads. So is this something that happens because of a migration of people, or is it something that happens because of a movement of the idea, but without people actually moving around? So do people just tell their neighbours about this new pottery type, or perhaps start selling, exchanging goods between different areas, or are they actually moving across? When we see a new material culture, what process does that represent? So the opinion has changed over the course of the 20th century about what the most important process there is. We started off with the assumption that it must represent a migration event, and then sort of from the 1970s onwards we start to see more ideas about cultural diffusion and exchange of ideas without movement of people. What we actually find in the case of the beaker phenomena, using the ancient DNA data, which is obviously a very good way of identifying whether people are moving, because we can look at the genetic signature of that movement, is that in fact both processes are occurring. There's both things are happening in different places at different times. So what this graph, what this figure here doesn't show particularly clearly is that concept. What we actually see is that for the most part, almost entirely in Iberia, where beaker culture originates, there's almost no movement in of the Yamnaya genetic ancestry that Selina was talking about. So the Yamnaya people have migrated from the steppe upwards through Europe, arrive in Central Europe, and then it seems that there's a diffusion of beaker culture without a movement out of Iberia, and then those people migrate on again. They acquire beaker culture and they migrate again across the rest of Europe and into Britain. So there's both an exchange of ideas followed by a subsequent migration event. How much impact does that migration event have in the UK? So elsewhere in Europe, we see some admixture, some mixing of Yamnaya heritage along with the indigenous Neolithic heritage that it replaced. But in the UK, at least, it seems there's almost total replacement. So this group of people living in the Neolithic farmers that were living in Britain towards the end of the Neolithic period, who in fact had just built Stonehenge within sort of a few generations of the before the arrival of beaker culture, are entirely replaced. And you can see this here using Y chromosome haplotypes. So we move from a sort of combination of I-based Y chromosome haplotypes in Neolithic Britain to R1b almost exclusively during the early Bronze Age, Middle Bronze Age and Late Bronze Age as well. So wholesale genetic replacement, which we can see both on the Y chromosome, that's the last slide, but we can also find it on the rest of the genome as well. So if we take individual genomes of individuals from the early Bronze Age, then we can, if we say that they can be made up of either Yamnaya heritage or Neolithic heritage, we find basically they're almost always Yamnaya, with very, very little, very small amounts of Neolithic heritage dotted through there. So it appears that there's a very significant population replacement event with very limited amounts of admixture occurring at the end of the Neolithic period and going into the Bronze Age in Britain. Okay. Phenotypically, we can also see some differences there, and some puzzling differences as well. So, puzzling lack of differences. So here, for example, looking at the HERC2 locus, which is one of the main ones involved in eye coloration, we can see that in Neolithic Britain, the frequency of dark, highly pigmented eye colour is quite high, so suggesting brown eyes, and that tends to, it seems, get somewhat lighter with the arrival of these beaker culture people. Similarly, for skin pigmentation, we see what we think is an increase in, or a reduction in skin pigmentation. So going from a sort of medium brown skin pigmentation to a lot lighter over the course of the beaker period. The one that's slightly puzzling is lactase persistence, so the ability to digest milk in adulthood, which is a very high frequency in Britain today. I mean, most people can digest milk in adulthood. There's retention of lactase persistence. There is lactase persistence. What we actually find, though, puzzlingly enough, is that it's still at a very low frequency all the way through into the Bronze Age. So it suggests that that very high frequency must occur somewhat later, going into the Iron Age, in fact. So it seems that there's been a very, very strong selection for lactase persistence, probably within the last couple of thousand years. And it's quite remarkable, if you actually look at the numbers on that, it must be one of the most strongly selected for loci that we've found in any genome anywhere. So the advantage of it must be enormous. Okay, so that was the first piece of work that we've done. More recently, within the last month, we actually stepped back a few thousand years more. So what I've been talking about there is events that happened within the last 4,000 years. Now we're going back to as far as 10,000 years ago to try to unravel another population change or cultural change which occurred in Britain. So this is the change from hunter-gatherer culture to Neolithic farming cultures, which occurred about 6,000 years ago. Britain is interesting in this respect because it's been debated for a long time exactly how this occurred. What we do know from the archaeology is that farming arrives in northern France and then stalls for almost 1,000 years before it makes the jump across the Channel and gets into Britain. And there's been various discussions about, or various ideas about how that process might have happened. One, which, as you'll see, is, had some merit to it, but has been discredited now, is that there's no migration of people, and just that eventually the indigenous people of Britain decided to switch from hunting and gathering to farming. Never seen that likely to me, but it has been discussed in that way. So the other ideas centre on migrations, but it's unclear really how many migrations or where those migrations might have come from. We know that there are various different farming groups present in northern France at the beginning of the Neolithic, and here's some ideas put forward by the archaeologist Alison Sheridan, where she suggests various movements from different parts of northern and central France, with an initial kind of movement up along the west coast of the UK, west coast of Britain, and then a later one up the east coast of Britain, and then a kind of subsequent one that goes just into Cornwall and southern Britain. Okay. So this is great because we actually have some testable hypotheses that we can work with here. So that bit's good. There are some proposals that we can test against. The downside is the nature of the samples that we actually have to work with. So the Neolithic period is quite a long time ago. It starts about 6,000 years ago. But nonetheless, there are a reasonably large number of archaeological skeletons, different collections around the UK that we could sample and work with. Interestingly, there are still sort of gaps in various places, which are probably to do with the geology, the underlying geology, which has led to the destruction of bone in those places. The Mesolithic period, however, is a lot longer ago. So we're looking here for samples that are perhaps as old as 10,000 years. And the extra few thousand years makes quite a big difference, it turns out. So almost all of our samples come from one set of locations here in southwest Britain, typically from cave sites, typically actually within maybe 50 miles of each other or something like that, including Cheddar Gorge and Kent's Cavern and sort of some other ones that are quite famous. There's one more set of samples from the Hebrides here, on a location called Carcoig. So one of the questions we can ask, I should have fixed that slide actually, so one of the questions we can ask at this point, we used a very similar kind of analytical methods than the ones that we used before. So we extracted DNA from the bones, we shotgun sequenced them, and we also did some DNA capture. We combined those data and then we plotted them out, as you can see here, on a principal components analysis. So one of the first questions we can ask is how different were those British Mesolithic hunter-gatherer peoples to the farmers that we find from Britain. And as you remember, one proposal had been that they're actually very similar. It's actually the same group of people, and then various other people have said, no, they're rather different. And what we find is that, yes, they are indeed rather different groups of people. So British Neolithic farmers fall here, which puts them in with a variety of other Neolithic farmers from elsewhere in Europe. But here on the PCO we have Mesolithic hunter-gatherers from various places in Europe. British ones are in green, and then various ones from mainly from Sweden, and also from sort of northern Europe, northern Spain, Luxembourg and so on, which is like the blue figures here. So rather different populations, which can also be differentiated in a variety of more robust statistical methods as well, not just by looking at all the principal components part. So it's a replacement, it's a population replacement, it's a different population. And who are those British hunter-gatherers most similar to? Well, they actually look rather similar. The one individual for which we have excellent data, which is the individual from the site at Cheddar Gorge, looks rather similar to individuals from Lochebourg in Luxembourg and Branchot, which is in France. They look rather different, somewhat different to the individuals from northern Spain, but there's probably some complex population history which best explains that. But yeah, the notable thing is that the individual from southwest Britain looks very similar to the individual from Luxembourg on the other side of the channel, suggesting that there's been recent migration during the Mesolithic that accounts for those two. Where do those Neolithic people that replaced them come from? I don't really want to get too far into the complexities of the F4 statistical analysis that we've used here. But essentially, what this looks at is we've divided our population data into different regions in the UK. So Wales, for example, here, England, at southeast, southwest England, central England, Scotland. And we compared those to individuals from Neolithic Central Europe and Neolithic Iberia. And so these are the two possible source populations, really. So do people come up along the Atlantic seaboard from Spain through France and into Britain, or do they come through Central Europe, up the Rhine and then into the Netherlands or into France and then across that way? And it seems that each of these different locations is probably best explained as originating from Iberia. And there is some weak evidence to suggest that perhaps different regions are slightly more associated with a combination of the two regions or not. When those Neolithic farmers arrived, did they admix to any degree with native hunter-gatherers? And again, I won't explain too much on this graph, but just to say it doesn't seem that that's the case. If we look at individuals from Wales on the western side of Britain, what we find is that they can be almost, that's this group here, they can be entirely explained as coming from the Iberian early Neolithic population. So they're almost indistinguishable from each other. Yeah? And if we look at England here, Scotland here, we find pretty similar stories, perhaps a little bit of admixture with native British Western hunter-gatherers, but not very much. So it seems that those Neolithic farmers arrive in Britain and don't really interact at all with the Mesolithic hunter-gatherers that they find there. And in fact, going through every individual in the data set using a program called Alder to look for the timing of admixture events, there's only actually, I think, 2 individuals, both from this site in Scotland, and western Scotland, Rashoili Cave, where we can estimate that within 4, 5, 6 generations, they had admixed with hunter-gatherers. That's the only example we have where that admixture is recent enough that it probably occurred in Britain. There's other evidence for more distant admixture that probably occurred before those individuals arrived in Britain as part of their ancestry. To what extent can we explain the data as being from a single migration event, and to what extent does it actually look like more than one migration? And again, if we plot out the extent of Western hunter-gatherer ancestry present in individuals that we find in Neolithic Britain, we actually find a bit of a climb So there's more of it in southwest Britain and less of it up here. And that seems to suggest that actually, well, one way of explaining that would be that there are actually two different migration events. One up into Scotland that moves downward, and one into southwest Britain that moves upward. Although I think my opinion would be that we are somewhat at the limits of the data here. We're really trying to squeeze as much information out as possible. Okay, so how are we for time? Shall I wrap up there?
Well, yes, but that's simple.
Yeah, or I can do another 5 slides on. Yeah, okay, right. Okay. So somebody nodded. So that's if you were hoping to get home early. I'm sorry. Okay, so one of the skeletons that we worked on was rather famous. It's the oldest and most complete. It's the oldest and most complete skeleton in Britain, which is a really sort of weird honour to have because there are both older skeletons and obviously more complete skeletons. So this is a near complete adult male in his early 20s who has been studied very extensively and dates to around about 10,000 years ago. He's on display now in the Natural History Museum as part of the Early Humans display, and he's a bit of a sort of one of our treasured display items. So we were able to, I think, was he not on display when we sampled him, or did we have to take him, we had to get him off of display, I think.
We took his head off.
Yeah. So Selina and our colleague Tom Booth took the skull, very carefully drilled it, a very tiny hole that's barely visible at all, certainly under the lighting conditions that it's currently held at, and we're able to extract enough DNA from the bone to actually get, we now have like a threefold coverage, 3x coverage genome, which is not great by standards from modern genetics, but it's pretty good for an ancient person of this kind of age. So Cheddar was actually excavated in the early part of the 20th century. The caves that he came from have been used as show caves, so they're a big sort of public attraction down in the southwest of Britain. So this was a postcard, I think, that was generated at the time with the two brothers who ran the cave. The cave's called Goff's Cave, and these are the Goff brothers who excavated it. This is, as you'd imagine, a rather posed photograph. And in fact, the skeleton, I think it took about another 30 years before all the bits were actually assembled and laid out properly, and they could actually work out how many parts of it were actually there. So there were actually, I've said it 2.3x, it's actually a little bit higher because we've been running more sequencing on that individual. And we have a lot of genetic information from that individual now. Because it's kind of famous, there's been various attempts to reconstruct what he might have looked like. This was the one from the 1980s, which is quite horrifying, isn't it? Quite disturbing. And what they've done here as well is included some kind of lesion, which seems to be on the skull, but actually it's a subject to debate whether that may have been incurred during the course of the excavation process, or whether it's actually a pathological thing that he would have had in life. So that was kind of the last attempt to reconstruct what this individual would have looked like. And during the course of this larger project that we were conducting, we were approached by a TV company and asked if we wanted to take some of the work that we'd done and make a TV show about it, make a documentary about it. And as part of that, the producer of the show decided that he would attempt to do a new reconstruction and compare how that kind of science has moved on. Facial reconstructions moved on over the last 30 years or so. Jump out, sorry. The thing that we were asked to contribute primarily was what genetic data could contribute to that facial reconstruction. And the main thing that we decided we could actually do was to make an estimate of skin colour, or skin pigmentation. And essentially what we were able to identify was that Cheddar Man probably had much darker skin than you would expect, certainly much darker than the previous reconstruction. And we did that by using a forensic-based method, or a method derived from the forensic literature, which takes a relatively small number of individual loci from across the genome. So the method uses 36 loci. We actually had 33 of those when we ran the analysis, and we had, I think we now have some evidence for all 36. So we identified what variance Cheddar Man had at each location, and then applied that algorithm. And the algorithm outputs basically it makes an estimate of which of these half dozen phenotypic categories your individual falls in. This is a scale known as the Fitzpatrick scale. And so what we came back with was this guy. And so it's been an interesting exercise for us. It's quite surprising to see this individual, not so much because of the skin coloration, but because I hadn't really realised he has a very small face relative to the size of his head. So he looks, I mean, it's quite unusual. It just looks really unusual, basically. Also, he has blue eyes, dark, slightly curly hair, and then this dark skin tone as well. Skin tone actually changes depending on which photograph you manage to find online. So he can look quite a lot lighter than this if the lighting's a bit less harsh. I think that was all I wanted to do.
Gracias. ¿Hay comentarios o preguntas?
I have a question. The blue eyes is something that you see also genetically? Because at the moment you say that the oldest Britons has like the pigmentation in eyes also was lost during time.
Yeah.
But this one was blue.
Yeah, and then that seems to be, completely goes and is replaced by this kind of brown eyes that come in with the Neolithic farming genotype, genomes. And then is brought back in with the Yan-Naya ancestry.
Yeah,
it is with the Yan-Naya. Yeah. So it's interesting. Yeah, it's kind of interesting. So it is quite cool, isn't it? Because you think of that blonde hair, blue eye kind of package all riding together, and that doesn't seem to be the case. And there are populations today who have that blue eyes and darker skin, but it's still, I mean, it's very unusual. Yeah.
No, si tienen una pregunta en español, también.
When you talk about that endogenous DNA percentage, what is a good amount in those kind of samples, and what is the rest of the DNA? Or what is...
Okay, let's start. I'll answer those two questions first, and then we can work on from there. Okay, so the most that we've ever had would be about 80%. And the way that it is measured seems to have changed, but you would not expect to ever get up to 100% because there are parts of the genome that you can't really map to. So you can't, these very short sections of DNA that we recover can't be unambiguously mapped onto some parts of the genome. Yeah, repetitive regions. So 80% is probably the highest amount. Most of the time, from the Mesolithic, of, I don't know, 40, 50 Mesolithic individuals we looked at, bits of Mesolithic skeleton. Yeah, well, think about all the early whole stuff and everything, yeah. We've got one at about 20%, which is Cheddar Man. We've got another one at about 10%.
Most others were 0.
Most of them are zero, yeah. Most of them there's no human DNA.
1340 with zero human
Yeah.
DNA.
From the Neolithic, you often get quite good DNA preservation, but again, most of the time you're looking at less than 1%. Well, actually, maybe most of the time, maybe 5% or something like that. Yeah, okay, yeah. But still, you know, it's still hard to justify sequencing those individuals up to a reasonable amount of coverage of the genome to actually do any useful work with those individuals. So, yeah.
Yeah, so what's a good percent would be what question do you want to ask?
Yeah.
How much data do you need to answer that question? If you just wanted to get a mitochondrial haplotype, then you just need the mitochondrial DNA.
Or a set. Yeah.
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 Then you don't need that much data. But if you want to ask bigger questions, then the more data, and then how endogenous it is, if that's good enough, it kind of depends how much money you've got.
Yeah.
Because you can just keep sequencing over and over and over and over, and each time get like 2% endogenous, and you'll keep making it more, but you've got to have a big bag of money to keep doing that.
Yeah, and
So
a big specimen, big piece of bone as well, actually.
Juan is relay or
Well,
thank you.
It would be good if I translate
Okay.
things.
Sure.
So, but do you think, like this last question, like shows the answers? Bueno, entonces, más o menos un poco retomando lo que estaba diciendo Ian, básicamente es que siempre es bastante difícil tener un buen contenido de ADN endógeno en una muestra, que lo que ellos mejor han tenido ha sido como 40%, 20%. Usualmente cuando pues no hay como un límite donde uno puede decir que una muestra es buena para hacer estos estudios de ADN antiguo, usualmente digamos que si tienen muy bajo ADN también endógeno del individuo, también va a significar un mayor gasto de las varias rondas de secuenciación que se van a tener que hacer como para ir enriqueciendo poco a poco esas secuencias que se están de alguna manera como digamos que se tienen ahí en la mira, pero normalmente pues se trabaja con lo que hay y también pues digamos que un 5%, un 6% es algo que se puede manejar y depende de la pregunta que se quiera contestar. Entonces para el ADN mitocondrial es mucho más abundante digamos porque hay mucho más ADN mitocondrial en una célula de lo que hay nuclear, entonces normalmente con muestras más degradadas igual se puede obtener una buena cantidad de ADN mitocondrial y se pueden hacer preguntas muy básicas si aún así se tenga digamos una cobertura baja de genoma.
Sorry, I hadn't finished though. There was one more part to that question,
Segunda pregunta.
which was about the bacteria. Yeah.
Okay.
So what is the rest of the sequence? We don't know really, but normally something like 10% of that, I think, those reads can be mapped onto some part of the bacterial database. So yeah, if you just blast the reads against NCBI, you'll get a variety of soil-based bacteria, typically. But the remainder, we don't find any matches for. Now that might be because there's such diversity in soil that we haven't sequenced that stuff yet. It's not on the database. Or it might be because those pieces of DNA are just highly chemically damaged during their time after the person died and before we sequenced them, or because they're actually some weird chimera of various bits of DNA that have been glued together during the sequencing process. So yeah, most of the DNA that we recover, most of the time, we throw away. This is no information.
Bueno, entonces, en esta segunda parte, lo que está hablando era acerca del contenido no endógeno de ese ADN que se extrae, y en su mayoría pareciera ser que es un ADN de origen bacteriano. Sin embargo, hay una gran porción de ese ADN que no logra, digamos, distinguirse muy bien, y podría ser en parte porque simplemente no se conocen estos organismos y también porque muchas de esas, digamos, las secuencias cuando se alinean pueden tener como características muy complejas, entonces puede ser simplemente que los algoritmos que se están utilizando para hacer esos, digamos, como de alguna manera, como los, digamos, las bases para poder hacer ese alineamiento no generen como una similitud suficiente para poder determinar qué es, pero en su gran mayoría parece ser ADN bacteriano, sobre todo el suelo en el que se han encontrado como estos restos arqueológicos.
¿Tú tenías una pregunta?
Sí, un poco técnico en español. Teniendo en cuenta la dificultad de la extracción de ADN, ¿cuál es la técnica de extracción que ellos utilizan para mejorar, pues obviamente, la obtención de este ADN?
So, like, considering the level of damage that the ancient DNA has, what's the technique, like more or less you can give an explanation of what's the technique you use for extracting the DNA?
Extracting the DNA?
Yeah.
Okay.
Yeah. So I've done it for about 5 minutes or something, yeah.
So when we use bone powder, we then we demineralize the bone. So we use, like, in part, it's a bit like using Kyogen extraction kits. So we do demineralization, and then we use silica spin columns to bind the DNA. The differences that we use for the ancient DNA, we make up all the different buffers and things ourselves. It's in a very sort of contained environment to not allow contamination in. But we use things like very large volumes of binding buffer, like 13 milliliters, to try and actually really make sure that you're binding everything to the silica spin columns. We do that.
That's probably the main thing.
That's probably the main thing,
Yeah.
It's small things, sort of repeating steps, leaving it longer, trying to make sure that we only use silica spin columns that have the shortest fragments, that we don't lose any of the shorter fragments. It's basically what we're trying to do. Because in a lot of the modern techniques, you don't care about the short fragments. So we're basically trying to just keep those shorter ones, and then, the main thing is the large volumes of binding both.
But what kit do?
So the main kit would be, so the spin columns actually come from, it's a Roche. It's a company called Roche, and we use them because they have, they go for they keep the silica as such that it binds the short fragments, but it comes with these big hoppers so that you can pour on top of the spin column these large volumes of binding buffer. It's a Roche large viral extraction kit, which we throw most away, but we just keep the spin columns is what we use from that, and then we make up the reagents ourselves.
So the main. Oh, sorry. Shall I stop there?
No, if you want. If you are just wrapping up the idea.
Well, okay. So the main differences that we have with ancient DNA, I guess, are that the DNA is very small. It's probably, on average, 50 base pairs, and then also there are some chemical modifications, which primarily the deamination of cytosines into uracil molecules. And for that, we actually do also do something.
Yes. We use UDG, uracil dehydrogenase, and what we're trying to do there is get rid of the uracils. So that basically, we use that before we do the library building process, and we do that so that it actually cuts. If it sees a uracil, it will cut there and remove that so that you don't accidentally pick up a T instead of a C because of this damage. If it's a U, it's cut, so you never sequence a T instead of a C. We can also do a thing called partial UDG, and when we do this, we actually treat the majority of the fragment, but the very ends of the fragment we leave. And so you can still get uracil at the end, and we do this so that we can authenticate the DNA as ancient. So when we sequence it and look at the ends and we see use, then we can go, great, we've got ancient DNA and not modern contaminant, which is really important when you work with humans.
Bueno, entonces esta fue un poco más. Pero bueno, básicamente la idea es que las técnicas que se utilizan no son tan diferentes a las que se usan para el ADN moderno, digamos. Simplemente que hay que tener en cuenta algunas cosas fundamentales. La primera es que como el ADN está muy fragmentado, hay que hacer esfuerzos adicionales durante los protocolos de extracción para asegurarse que esos fragmentos pequeños sean rescatados y no se pierdan. normalmente con el ADN moderno uno no se preocupa mucho por los fragmentos cortos, porque probablemente son cosas que están medio degradadas y uno necesita es como el DNA de buena calidad. En este caso uno lo que está tratando es de recuperar tanto ADN como sea posible. Bueno, y adicionalmente hay una serie de cambios químicos que también se producen en el proceso de degradación post-mortem del ADN, entonces hay que aplicar otros métodos. En este caso, por ejemplo, el cambio más común y más complicado luego en los análisis de datos es el hecho de que las citocinas se convierten en uracilos. Entonces la idea es que agregando un reactivo que permite romper los enlaces donde están los uracilos y extraer esa molécula, de alguna manera se está evitando que luego haya una confusión en los análisis cuando en la secuenciación en vez de tener una C se tenga una T, pues en vez de una citosina sea una timina. Pero a veces lo que se hace también es que esos patrones de aminación que generan el uracilo también ayudan a diferenciar cuál es ADN antiguo del ADN moderno que pueda estar contaminando la muestra. Entonces muchas veces lo que se deja es como que parcialmente se remueve parte de ese uracilo que se ha producido, pero también se dejan algunos cuantos fragmentos, de modo que en los procesos bioinformáticos posteriores de análisis de la muestra se pueda definir cuál es antiguo y cuál no.
I don't know what you're looking at. It's
¿Otra pregunta o comentario?
Yes, I have a question. I don't remember in which of the cases that you exposed was this graph when you showed the Y chromosome haplogroup replacement over time. That's only based on Y chromosome inheritance, but you have data based, for example, in mitochondrial haplotypes, because maybe the rates of maternal, paternal inheritance are different. I'm not sure in this case that could be observed from the data, because I know this is a specific migration case, but some studies suggest in other scenarios that women migrates longer distance than men. So I don't know if from this kind of data you could observe some that kind of variations in the observed migration from male populations and female populations in their replacement.
Now, you'll notice that Selina is looking at me, and
Yeah,
I
I
infer
didn't know if
from
you were
that.
about to talk.
No.
No, I would.
Have you got it? Right, okay.
Yeah, well,
I couldn't remember.
Well, no, because we do actually look at the whole genome as well.
It was just translated question.
Okay.
Bueno, para todos básicamente, la cosa es que, bueno, la evidencia que se está mostrando del reemplazo de los haplogrupos del cromosoma Y, bueno, lo que pregunta Nicolás es si además también se ha evidenciado si el caso es semejante para la mitocondria, que estaría representando pues los linajes maternos, o si realmente hay algunas diferencias en esa migración. Pues eso es como en resumidas cuentas lo que estás preguntando. Perdí a Brace.
Yeah. Sorry.
Yeah.
So what I was going to say, so although we showed the Y chromosome data there, the same pattern is reflected when you look at the whole genome. So we use that because people have looked at male chromosome movements before, or Y chromosomes moved before, so we show that and have that in the paper because it fits then with the pattern that other people have done. But when we look at the whole genome, so all the SNPs throughout, it reflects the same thing. But if we hadn't done that, that would have been an entirely, I mean, it is a valid question, but because the whole genome suggests that it's not.
I have a feeling that you can't pick it up on the mitochondrial DNA because there's already too much sharing of mitochondrial haplotypes between Britain and the rest of Europe. But if that wasn't the case, then we probably would have already picked it up because a lot of mitochondrial work has been done on archaeological humans in the past.
I was just thinking about Neanderthals as well, because initially when they looked at Neanderthals and just looked at the mitochondrial DNA, then they thought there was no interbreeding with modern humans. So it's like you're saying, they got the wrong picture, and then when they looked at the whole genome, they went, Oh,
Yes.
yes, they did.
Okay.
So it's very important to look at both and not just look at that in isolation, as you say. Yeah, it changes the picture.
Bueno, otra vez para resolver. Pues en teoría, claro, es un escenario muy común el que describe Nicolás, donde la mitocondria puede estar contando una historia muy diferente a la que nos está contando el cromosoma Y, pero en este caso, como los análisis también se hicieron con datos autosomales, o sea, con muchos cientos de miles de SNP a lo largo de todo el genoma, eso demostró que el patrón era completamente igual, es decir, que las migraciones habían sido masivas de estas poblaciones y no había como un sesgo a nivel de hombres o mujeres migrando en cantidades diferentes.
Olguita, y dos comentarios más para terminar.
Okay, those migrations policy are very interesting for the UK. And what about the other countries in Europe? Or is that something that needs to be done?
Yeah, go on. Yeah. Sorry. Yeah.
So actually, so looking at like the big period, that was all done in Europe beforehand, and when they look at migrations across Europe, it is actually slightly different in Britain, because but at the end that Ian was showing, where there's no integration and admixture after, so those the farmers arrive in Britain and they their signature takes over from the Mesolithic people, then there doesn't seem to be any interbreeding or admixture between them. It's very separate. Whereas when people have looked across Europe prior to that, there is a lot of interbreeding and admixture that goes on. It's slow, but there does seem to be a lot more admixture, so it is different. And we don't really know why it was different in Britain, but a possibility is because by the time people had been moving across Europe, by the time they get to Britain, this end point, they've actually already really established farming, they know what they do, so they're not really interacting with anyone else. By the time they get there, it's kind of...
I mean, there's some evidence that there's
so,
nobody there.
Yeah, exactly. Yeah. The second one is that there was just no hunter-gatherers left, or very few, so there wasn't really anyone to interact with.
But one explanation for, so this is the Greek Neolithic in red, Greek Neolithic individuals. These are German Neolithic individuals. So and this is Britain, and I can't remember where the magenta ones are from, kind of more Central Europe. Yeah. So one explanation for why there's this shift is that we're seeing increasing amounts of hunter-gatherer admixture as they make their way across Europe. But then by the time they get to Britain, then we don't have any more admixture. But that's been one explanation for why it seems that the Neolithic farmers are kind of creeping on the PCA plot towards the Mesolithic hunter-gatherers.
The one place we're missing is France. We don't have much data from France.
No.
So we would like data from France if you want.
That material has not been available for DNA analysis. But I believe that there are now groups that are working on it.
There are dead people from France.
There are certainly dead people from France. There is a paucity of early Neolithic, kind of middle Neolithic skeletons from northern France. Which it means we can't make a direct comparison between the most likely source population and Britain. And that's because of the geology. So there's just no skeletons in that region.
Maybe the French are doing it, no?
Maybe. Hopefully.
Yeah.
I hope so.
Yeah, either French groups or French groups in conjunction with other groups, yeah. Yeah, I think it is now ongoing, but there's just been a slight delay.
Bueno, esto también tiene bastante, entonces...
Sorry.
It's like a memory test. Yeah.
But I'm getting good at it. Bueno, no, la cosa es que en las otras poblaciones europeas hay muchos estudios, excepto Francia, que bueno, ya ahorita explicaremos un poco más, pero básicamente lo que se ha mostrado es que el caso particular en Inglaterra es que parece no haber habido tanta mezcla entre los que llegaron, los granjeros, los agricultores que llegaron y los cazadores recolectores que estaban ahí. Entonces hay algunas ideas. Una de las ideas es que simplemente no había una densidad de población muy grande de cazadores-recolectores al momento que los otros llegaron, y lo otro es que probablemente no interactuaron mucho con ellos porque ya tenían sistemas de agricultura como mucho más establecidos, mucho más desarrollados. Igual entonces también lo que nos estaba mostrando Ian en los PCAs es que básicamente se ve cómo fue aumentando la mezcla con los cazadores-recolectores durante todo ese trayecto. Entonces seguramente mucho de ese, digamos, de esa herencia que se puede dar de cazadores-recolectores en esos granjeros que luego reemplazaron las poblaciones en Inglaterra, realmente viniera ya con ellos desde como estas otras poblaciones europeas. Y bueno, y ya el caso específico de Francia es que, pues primero, por cuestiones geológicas hay muchos lugares donde no se ha preservado estos restos arqueológicos, pero también hay algunos asuntos más políticos en el hecho de que hay mucha más dificultad para hacer investigación en genética humana y todo esto. Entonces probablemente ahora ya haya gente haciendo algunos trabajos como dentro de Francia y eso, pero ha sido mucho más demorado que en muchos otros países europeos.
¿Una pregunta más? ¿Comentario? Ian, Selina, thank you so much. Muchas gracias a ustedes por acompañarnos y los esperamos en 8 días con otra interesante charla. Gracias.
Pero igual yo lo que es como de llamar porque, perdón, es que, o sea, igual, ¿qué te iba a decir? O sea, yo tengo que estar en el aeropuerto antes de las 7 de la noche, o sea que también es porque mi vuelo es a las 7:50. Tengo que registrar. Sí. No, es a las 7 de la mañana. Tengo que...
Right, okay. Right, okay. Right. And where is the material from? Places.
Well, the most oldest is only one place
en Perú. Hola, Pedro. Muy bien, digamos, qué pena que no te haya podido comunicar lo que estoy diciendo en internet. Pero sí me regaló ahí. Vale, vale, yo igual voy con ellos y de todas maneras yo creo que sí te van a pedir el favor. Porque tengo el vuelo en 2 horas.
Vale, so it's easy to miss the key part of the Petrous Bone if you're drilling into it. Yeah, whereas teeth I think are a bit more unique.
Sí, porque pues ella me decía en esas películas como que ellos que son como en Latinoamérica y cuando tú lo miras ya aparece como los fragmentos de ADN y en Norteamérica o Europa es como súper
Es que somos muy recientemente mezclados entre cosas muy diferentes, por eso es que se resalta tan fácil. Claro que también está súper mezclado.
Chao, Anillo. Cuídate.
I do, yeah. Let me look.
es más africano. Yo creo que en el suroccidente hay muchos científicos. Recientemente hubo una migración japonesa enorme.
It's on display, yeah. It's on display, yeah. But the reconstruction kit is is sitting in a box, waiting to go on display. So yeah, but hopefully the second one, yeah.
Se hace, es una empresa que se llama, creo que es canadiense, claro, no la hacen en Colombia, porque el colombiano no está dentro de los modelos, pero si tú tienes a alguien en Estados Unidos que puede hacer la gestión.
No, yeah, it's sitting in a plastic box in the office next to it. It's really scary because it's just like this clear plastic box.
Sí, y además, pues te sacan muchas cosas de salud que no sé, pero son los rasgos. Sí, pero después yo, pues son poquitos, pero rasgos y lo de ancestros. Y por cromosoma te sacan otra empresa. Well, it's not
Muchas gracias a todos.
Thank you. Yeah, that's good. Yeah.